communities. And but before starting, I wanted to say some words about Jena University, because Jena is not so well known, although it's quite a traditional university, and there were famous people working at Jena. For example, Ernst Haeckel, who came up with the biogenetic rule, meaning that ontogeny is recapitulates phylogeny, and he also drew these nice art forms of nature. So it's a big volume of many colorful and artistic paintings of organisms. And then also Matthias Schleiden worked there. He discovered the living plant cell while he was in Jena. He also was at other universities. And yeah, there's a relation to Great Britain or to England, because the dead plant cells were discovered almost 200 years earlier by Robert Hooke. Unfortunately not in Cambridge, but in Oxford. And yeah, then my usual starting point is optimization. So I say that due to the Darwinian theory of evolution, certainly the concept of optimality plays an important role, but probably it's not necessary to tell it here, or maybe even counterproductive, because all of you know that often biological systems are not optimal, exactly due to phenomena which we can describe by game theory, like the prisoner's dilemma, where the suboptimal solutions occur. But I think there are some properties of living organisms which can really be explained by optimality principles. For example, just to mention one thing, optimal hematocrit theory, where the optimal hematocrit, meaning the optimal volume fraction of red blood cells, can be computed based on maximizing some objective function. In that sense, in that case, the maximal oxygen transport rate. Yeah, and as you know, evolution is actually almost always co-evolution. That means several species evolve together and they interfere with each other. Yeah, and this is actually, I think, the main reason why often optimal systems are not, optimal properties are not reached, because other species or other cells interfere. And yeah, so that we have a game, or we also call it conflict or competition. And yeah, this often, but not always, involves communication. So I think communication is an important aspect which is sometimes neglected in game theory, because sometimes or often the Nash equilibrium depends on whether there is communication between the players or not. Yeah, and main topic in my group is for many years already modeling of metabolic networks. And for metabolism, there are several thematic principles have been suggested. I think the two major ones are maximum molar yield and maximum flux. So, and molar yield means the stoichiometric ratio, in a sense, between product and substrate, meaning, for example, how many moles of ATP can be produced per mole of glucose, or how many moles of an amino acid. And this is mostly a stoichiometric property, so it can be calculated just from stoichiometry, and maybe a little bit thermodynamics, because one has to include also the information which reactions are reversible and which are irreversible. And and then the other criterion is maximum flux. For example, maximum ATP production. And sometimes this is confused in the literature, but the important difference is that maximum flux means how many moles per time, whereas yield means how many moles per mole of substrate. And in many situations, these two criteria contradict each other. Sometimes they are in agreement with each other, meaning higher yield means higher rate, but often they contradict each other. And a very striking example is energy metabolism in cells when we compare fermentation with respiration. Yeah, fermentation has a low yield. Fermentation essentially means glycolysis. The terms are slightly differently used in literature, but here I mean really from glucose to, for example, lactate or ethanol or acetate. And yeah, this gives 2 mole of ATP per mole of glucose. I think in this case of acetate it gives 3, but very low number, but a high ATP production rate. Yeah, for example, in striated muscle, or in our body, we often use glycolysis. In higher organisms, it's usually not called fermentation. So fermentation is the term for microbiology. Under heavy exercise, muscles use mainly glycolysis because of the high ATP production rate, ATP per time. And respiration has a high yield, depending on the organism and also on conditions. It's 15 to 30 moles of ATP per mole of glucose, but a low ATP production rate. And I think the mechanistic reason why it's so low is first because the pathway is much longer. So it involves glycolysis, the TCA cycle, and oxidative phosphorylation. And also the proteins, especially in oxidative phosphorylation, are in the membrane, so they are confined to a 2D space, whereas glycolytic enzymes are cytosolic, so have a 3D space. And yeah, and this can be described by a game theory, and actually this was Thomas who came up with this idea, so I worked a lot together with Thomas Pfeiffer. We know each other for 16, 17 years from times in Berlin, when we both were in Berlin in Germany. And yeah, I think also you can probably tell better, but I think you heard lectures at classes with Peter Hammerstein in Berlin, who is a famous game theorist, and also some of these ideas trace back to Peter Hammerstein. And yeah, and the idea is as follows. So we have the two pathways, glucose to, let's say, ethanol in baker's yeast, and respiration. So the two pathways overlap partly, and then here's TCA cycle and oxidative phosphorylation. Yeah, probably this number should be corrected downward, because now recent numbers in our body are it's about 30 ATP, and in yeast it's even lower, but still higher than in glycolysis. And and to give a number for the rate, as in striated muscle, fermentation, glycolysis is about 100 times faster than respiration. So, and now when two cells...
so is that the flux or the rate of ATP production?
Yeah, I don't know. I've actually looked up, it's from a textbook by Stryer, and it doesn't say whether this is the rate of glucose consumption or of ATP production, yeah, because this makes a big difference. But I think in either case, the ATP production rate is higher, because even if you divide 100 by 30, it's still higher. But this is an important point. I should look up the original literature for that. But yeah, it's. So, and if two cells or two species compete with each other, each of them has two strategies in a sense, namely respiration or fermentation. Yeah, and then the choice of strategy could, for example, be done by epigenetic regulation. Of course, it's not a rational thinking. It's in a sense by the strategy is chosen by selection, and it's a game theoretical problem, because the outcome for each player depends not only on the choice of its own strategy, but also on the strategy of the other one. Yeah? Because they share, it's assumed here that they share the same pool of resource. Maybe on a grape, for example, there's some glucose, and then several microorganisms compete for each other. And if one of them switches to fermentation, it uses up the resource faster and takes it away from the competitor. And yeah, and this can be described in several different ways in game theory. Actually, in this paper, together with Thomas and Sebastian Bonhoeffer, we described it as a tragedy of the commons, which is actually an n-player game, which it is actually, because there are many species. But maybe for didactic reasons, we could also write it as a two-player game, so and then use the traditional payoff matrix. And so we have the two strategies, respiration and fermentation, and then here the payoffs for the first player and for the second player. So it's a symmetric game, therefore this is then the opposite of this one. And zero means that the population dies out, because the pure respirator would die out in competition with the fermenter, or actually respiro-fermenter, because often a mixture is used. But anyway, so as here we, I didn't give the source. This is a paper we published later, where we described it as a prisoner's dilemma, and we used the stationary population density as the payoff, which might be questionable whether this is a good payoff measure, but it gives at least a nice prisoner's dilemma, I think.
Can I ask a question?
Yeah.
So if you talk about the steady state population density, there's also an underlying dynamics?
Yeah, exactly. Yeah. So so we described it by a differential equation system, which is not shown here, where we have three variables, namely, we have a constant input of substrate, and then the substrate, the first variable is the substrate, because the faster it's used up, the lower it goes at a stationary state. And then the two population densities of the two populations. Sorry?
A classic chemostat-like model.
Yeah, it's similar to it. It's not exactly the chemostat because there is no dilution flux. So the populations can die out, or can die. Also there is a growth and death process, but there is no dilution. So, for example, substrate is not washed out. So in the chemostat, also the substrate is washed out. Yeah. But it's similar to that. Yeah. And probably, I think we never did it, but it could also be written for a chemostat, and then probably very similar results would come out. And the interesting thing is that, like in the prisoner's dilemma, the Nash equilibrium is worse for both of them than this situation. Yeah? This is sometimes called the Pareto optimum. So if both respired, then they were better off because they would use the resource more economically. But there is a temptation, so to speak, to switch to a fermentation because then the fermenter has an advantage, can grow faster, can outcompete the other one. But since both of them has this temptation, they end up in the Nash equilibrium that they all ferment.
Can you just remind me again how much more efficient or how more rapidly fermentation produces ATP compared to respiration?
Yeah, it's about 100 times faster. I think also in our paper we used figures like that. So two orders of magnitude.
So significantly greater than the payback if you were.
Yeah. But this is of course an important.
There, right?
Sorry?
It wouldn't enter in that equation because if you assume to be respiration, you die out and it doesn't matter.
No, I think it really matters. If the ratio between the flux of respiration and fermentation were lower, as if they were nearer to each other, then there would not be a prisoner's dilemma. Okay. So we analyzed it later in another paper, so we showed that it could also be a so-called harmony game. Mm-hmm. So in harmony, everyone would respire. This is really, the prisoner's dilemma only occurs if there is really a large difference in rate between respiration and fermentation. Yeah, and then of course it's interesting to look at the experimental observations, and there are actually several yeast species. The most famous one is baker's yeast, but also Saccharomyces bayanus and Schizosaccharomyces pombe and others use fermentation even under aerobic conditions. Yeah, and this is the interesting thing. Yeah? Even if there's oxygen available, if sufficient glucose is available, they use mainly fermentation or respirofermentation, meaning that they use respiration until saturation of the enzymes, and then in addition they use fermentation. And I read that Deckera bruxellensis is used for fermenting the Belgian beer. So it's not always done with baker's yeast. And then there is another group of yeast species that are non-fermentative, so they rely on respiration. So if there were no oxygen available, they would die out. And then another one are capable of using fermentation, but do so practically only under anaerobic conditions. So in a sense, these two groups behave in a cooperative way, whereas these in a competitive way. So actually, they correspond to the Nash equilibrium in the prisoner's dilemma, whereas these, in a sense, either they have other parameter values so that are in the harmony game, or they have somehow managed to escape from the Nash equilibrium in the prisoner's dilemma and could do the transition to cooperation. There could be different explanations, either spatial structure or stochastic effects. So we also did some stochastic, or Thomas did some stochastic simulations, showed that this could lead to cooperation. Yeah, and actually,
another question. Have you also looked what happens under fluctuating conditions? Because this is at steady state, but under fluctuating conditions might perhaps be advantageous to be.
Under varying conditions, yeah. You mean? So I did not. Have you done something on?
I mean, depends if you assume like a normal life cycle where you have batch grow in there exponentially and then you transfer to the next batch, it's something where rate would matter.
Yeah, or where the glucose availability would vary in time.
Yeah.
Then I can imagine that it makes, in some cases, more sense to be able to store a lot and then slowly respire.
Yeah, if you're in a dynamics where you actually can store, but that requires pretty specific.
Because many of these yeasts can store large amounts, right?
I don't know. I don't know.
So trehalose,
but
what is the storage substance? But this could be a possibility.
Well, they effectively store the energy in ethanol that they then respire, right?
Yeah, this is of course an effect which we neglected because, in a sense, because they excrete ethanol, but then there is the deoxic shift. So if glucose is exhausted, then they switch to respiration, take up the ethanol, and respire it.
So they're not losing all that carbon.
So this is not completely lost, but still something is lost because it has a lower ATP yield as if they were directly respiring it, because at some of these steps, at least one ATP is lost.
Yeah, from acetaldehyde to acetyl coenzyme A. ATP.
But it's still, the net loss is only very small.
It's very small, that's true. So in a sense, this description only applies to the first phase.
I have also a question. And for these two groups where the species are cooperating, is there any difference in the enzyme kinetics? I mean, is there perhaps not so much of a difference between this speed difference that we've been?
I must confess, good question. I must confess we didn't look at these other species. I just sorted out these species, but we didn't look at the kinetic parameters for them.
Perhaps the difference gets smaller and smaller, so you have to switch to a different strategy.
Yeah, but I would doubt that because I think in most species and most cell types, respiration is really slower than glycolysis. Actually, the same effect can be observed in cancer cells. Yeah? There's the famous Warburg effect. Warburg observed, as the German biochemist, observed that cancer cells mainly rely on glycolysis. And then for a long time, the people thought that this is because of lack of oxygen, because if the tumor grows, it doesn't have enough oxygen. But now it's becoming more and more clear that even if there's enough oxygen available, there's this Warburg effect. And I think the reason is that because of the higher ATP production rate. And this shows also that glycolysis is much faster, because the pathway is much shorter. So I would think that also in these yeast species, glycolysis is much faster. So that it still is a prisoner's dilemma, but for some other reasons, these species have evolved towards cooperation.
But they employ the most common strategy, right? The Cerevisiae is the weird organisms. The vast minority of organisms.
There are also several others, but probably the most of them. For example, in E. coli, it's also observed. That's often called the overflow metabolism. Also if there is, then it depends on the glucose. Now here also depends on the glucose concentration. If glucose is high, then glucose is not completely respired down to carbon dioxide and water, but some of it is excreted as acetate. And then biochemists often, or microbiologists often call this the overflow metabolism. So they say there's some overflow. But I think the term is a bit misleading because it's not really an overflow. It's really, in a sense, a good strategy by E. coli to produce ATP as fast as possible.
But your feeling would be that the Crabtree positive yeast are the majority or minority?
Minority.
Because, I mean, we really have difficulties to find good Crabtree negative ones. I mean, in your isolates actually, so
right.
Which is probably because they're coming from grapes where you would have a bias towards
maybe
Crabtree positive. But I mean,
it would be a revelation if it turned out that most microbes associated with sugar-rich niches were in fact Crabtree positive.
I mean,
because most people would assume the other way around, right?
I don't know. I don't have a very good feeling for that right now. But like if I mean the Peace Corps papers, they anyway describe the whole thing as a more gradual thing, like in terms of what conditions you need to start fermenting, and how much you ferment if you do fermentation. But it's from their sample of yeast, which is like 150 million years, so from Cerevisiae down to the Cucurbitacea approximately, which branched about like 150 million, I think, years ago, in that range. And in between, there isn't that much in that lineage. So I mean,
that we know about.
yeah. So I mean, yeah. So one has to go further back in yeast diversity to look at. So we really, I mean, I would like to have a good Crabtree negative yeast that is as closely related as possible.
Yeah.
But then we don't really have that.
Yeah, maybe to explain what Crabtree positive means, there's the Crabtree effect called of a biochemist, meaning as a Crabtree effect means that under high glucose, these yeast cells downregulate respiration. It's even so that they downregulate in order to increase fermentation even more. And this is done here. And I'm not sure, are both of these groups Crabtree negative? Or
I say only respiration, then.
Yeah, here this is negative. Yeah, here this has no meaning because they have only respiration. They are non-fermentative. Yeah, and this actually led us then some years later to a critique of flux balance analysis. But I mean, this is a long story. I can now explain it only briefly, but maybe later we can discuss it in more detail. Because there is also a debate what is actually maximized in flux balance analysis. Because most people work in FBA claim that growth rate is maximized. For example, Bernard Paulson, in many papers, it's written growth rate is maximized. Although if you look in some earlier papers by his, so from 93, 95, he wrote that yield is maximized. So I don't know how this shift came about. And I believe that what is actually maximized is molar yield, because this is because FBA is a stoichiometric approach. So they look at the stoichiometry of the network and include information about irreversibility of reactions, but there is practically no kinetics, and therefore I can't believe that growth rate is maximized. And and now if you now look at the experimental observations for example, if you compare respiration and fermentation, then FBA would predict respiration, because it gives a higher yield rather than fermentation. And actually, this was also written in some papers on FBA that they had really difficulties to predict the fermentative pathway, and then they had to include some site constraints, for example, limit the oxygen consumption rate. And for me, this looked always a little bit like tinkering with the methodology until the desired outcome comes out. Therefore, actually Thomas, myself, and David Fell, who will come later to this program, wrote this paper where we actually criticized some of the assumptions of FBA. Also we still think that FBA is quite useful in some applications, for example, to look whether some flux distribution is feasible at all, but and then also in biotechnological applications to look which yield can at best be realized by gene manipulation. But we think that the flux distribution this is observed in evolution, or in other words, we don't think that evolution favors, as the title suggests, that evolution favors pathways with high yield, but rather pathways with high rate, which is in a sense could erroneously be derived from the claims of the FBA, because they often speak about rate, but this is not really what is done in the modeling approaches, as far as I understood it. Yeah, then I would present you another example from microbiology, namely extracellular enzymes. I have already understood from the discussions over coffee that most of you know these examples. So there was a famous experiment by Greg and Travisano, I think in London, in England, where they took also baker's yeast and then silenced or even knocked out the suc genes, this is sucrose genes. These are the genes producing invertase, and invertase is the enzyme hydrolyzing sucrose into glucose and fructose, and then actually there are two forms, namely the intracellular and an extracellular form of invertase, and if the extracellular form is no longer available, then sucrose cannot be hydrolyzed outside of the cell, and then they mixed the wild type with the knockout cells, and then of course the knockout cells are a sort of cheater. Yeah? They don't produce extracellular invertase, but can use the glucose produced by the other cells. And then they plotted here the relative fitness of the defector cells as a function of what they call sociality, which is in a sense the cell density. So at high cell densities, the defectors have an advantage, and at low densities, the cooperators have an advantage. And then, as the title suggests, they called it the prisoner's dilemma. However, we were skeptical about that because in the prisoner's dilemma, defection is the only stable strategy. There's only one Nash equilibrium, while the observations of these authors show an advantage of cooperators at low cell densities. And so we asked the question whether this is really a prisoner's dilemma, and we think the question is relevant, for example, in biotechnology, because actually extracellular enzymes play a large role a major role, for example, in biofuel production. Yeah? When corn or some waste of plants is digested to produce ethanol, then the cellulose must be hydrolyzed, and this can only be done by extracellular enzymes because cellulose is a macromolecule that cannot be taken up in the cell. And then these cellulases and hemicellulases are extracellular enzymes. And then we in a review where Thomas was also involved, and Jan Kreft, who will also come to this program, we suggested that it's rather a snowdrift game, which is also known as the Hawk-Duff game. And in the Hawk-Duff game, there are two Nash equilibria of the main diagonal, and this allows coexistence of cooperators and defectors. And this was then also experimentally and theoretically shown in this paper. Actually, they were faster than we, because we had at the same time we also worked on that topic further, and anyway, we did it only theoretically, and they have nice experiments, and then they showed, and independently we also had the same result, that it can be a snowdrift game, but depending on parameters, it can also be a prisoner's dilemma or a harmony game. It depends on the benefit to cost ratio, also the cost for producing the extracellular enzyme, and then the benefit of that. For example, at low cost, as if it's very cheap to produce this, then it can also be a harmony game. And to explain harmony game, this has only one Nash equilibrium, but they cooperate, whereas prisoner's dilemma has defect, in which, and examples, for example, are animals forming groups to protect against the cold or against predators, like penguins, and there is no temptation, no incentive to switch to another strategy, because if one goes out, he will freeze, and so the only Nash equilibrium is to cooperate.
So in yeast, how would that work? That it would be advantage to have when we expect
I will come to that. I show that. But before that, I wanted to show a nice classification of two-player, two-strategy, as a symmetric two-player, two-strategy games. So this is actually the simplest case in game theory, but still there are quite a number of different game types. We took it from this paper. So then here we, I should tell, these are the variables for the payoffs. And since it's this symmetric game, you should imagine it's R/R, player for the first player, for the second player, then S/T, T/S, and PP. And then they did a normalization because, of course, the Nash equilibrium does not depend on on scaling, neither on adding a constant to all the payoffs. Yeah? Therefore, we can shift it and also normalize it. However, there is one condition, namely we assume that R equals 1 and P equals 0. And in some cases, there can be degenerate cases where both of them are 0, or in some cases it's turned around so that P is larger than R, and then, so to speak, the entire coordinate system has to change. Therefore, sometimes if we play with the parameters, then there is a switch, which is not so easy, but it can be analyzed, of course. And so, and then, yeah, we have the, for example, the prisoner's dilemma, where the temptation is quite high, as is actually the highest payoff, but the S is the lowest one, the so-called sucker's payoff. And here is the hawk-dove game, leader game, battle of the sexes. Actually, there are even 3 different types of harmony games, coordination game, and so on. And yeah, then the question arises, why can there be coexistence between cooperators and effectors? Because usually one would think that effectors always have an advantage because they save the coast for producing and secreting the extracellular enzyme. And as in many other games, we think that spatial effects here play a major role because the cooperators, they excrete the enzymes, which here are represented by scissors, and this cuts the sucrose into glucose and fructose. And then locally, there is a higher concentration of glucose because of the finite diffusion velocity. So the effectors, they save energy, but they have a lower local glucose concentration. And yeah, and then of course we should describe it as an n-player game because there are many cells. And there are actually 2 possibilities of treating large or even infinite populations. Namely, we could decompose the game into consecutive 2-player games. This is often done for higher animals. Yeah? For example, if male deers in a forest meet each other and fight against each other, then these are consecutive games. And there's a so-called Bishop-Canning theorem where you can calculate the equilibrium frequency, for example, of the hawks and of the doves, from these payoffs R, S, T, and P. But strictly speaking, this is only possible for consecutive 2-player games. Actually, in this paper by Gore and van Oudenaarde, they use this theorem, although it's not strictly justified because yeast cells interact every cell with every cell, not consecutively. And the other possibility is so-called playing the field, where each player interacts with all other players simultaneously. And I will show you how this can be done. Also for this concrete system, we expressed the growth rate by a Michaelis-Menten or Monod equation as a function of glucose. And then the cooperators have coasts, so this should be subtracted. However, they have a locally higher glucose concentration, and therefore, if S is higher, so this is substrate concentration S, inserting this here, we see that also, of course, the growth rate is higher, and so therefore we have a negative effect and a positive effect, and at equilibrium, they are equal to each other, so that at equilibrium mu c equals mu d, the factors, and and then it's important, this curve was not at the equilibrium situation, but this was the initial rate. Craig and Trevisano measured the initial growth rate, and then it's very easy because then we just divide these 2 terms, so they need not be equal, just the ratio, and then we came up with this nonlinear curve, whereas Craig and Trevisano fitted it by a linear curve. And I think the nonlinear curve is better because it also describes the right asymptotic behavior, because for very large density it must be constant, and also at very low density, because then at low density means there are isolated cells, so to speak, and then it doesn't matter whether they are 1 meter or 1 kilometer apart from each other.
What about the point at the top?
This one?
No,
No, higher.
up.
So yeah, I don't know. This probably an outlier.
It should have been discussed.
I think it's an outlier. Yeah.
Or explain.
And so and this was then the paper where we published, where we also had these three cases and these three game types, and then we computed the equilibrium frequency. This could even be done analytically. So, and now three cases occur, and this is so here we equate these two terms and then solve for x for the frequency, giving here the equilibrium frequency. But now there can be three cases, namely x asterisk can also be larger than 1, hypothetically, but of course it cannot be because a frequency must always between 0 and 1. But then it can be shown by differential equations that it tends towards 1. So it tends towards 1 and it stays at 1. So this is then all cells cooperate. And this is the case when the benefit to cost ratio is large, or cost to benefit is low. So if it's very cheap to produce the extracellular enzymes, then all cells will do that. Then in the intermediate case, we have a coexistence, meaning there's a Hawk-Duff game. This is harmony game, Hawk-Duff game, and this is the prisoner's dilemma. If it's smaller than 0, then we have to put it equal to 0, so all cells defect. And now it depends. In the invertase case, the cells will probably survive because there's also an intracellular invertase. But in other cases where there's only extracellular form and the cell is really dependent on the extracellular source, then the population can die out in these cases, which is sometimes the case that the population gets extinct because the cheaters take over, so to speak. And the reason why we don't observe it now is maybe because they have died out. This can also be shown in a graphical way, depending on cell density and the coasts. For low coasts, this is the other way around. So here are low coasts, higher coasts. At low coasts, we can have, for example, here the harmony game, then Hawk-Duff game, and prisoner's dilemma. And the dependence on cell density is also intuitively clear, I think, because at high density there are so many cells, and if at least some of them produce invertase, all the others can benefit from them. Whereas if it's low, then more cells need to cooperate. And then an interesting question is, what is the optimum in the biotechnological setup? Let's assume we can manipulate the cell density, at which point there is maximum productivity of, for example, glucose, or in biofuel production of finally ethanol. And then we have to distinguish between volumetric productivity and, I think, cell productivity, meaning productivity per volume or per cell. And if we take the volumetric productivity, then we get this curve. And here it's constant. However, if we now also want to, in addition, maximize cell productivity, then this point is the maximum. And we called it the optimum at the edge of harmony, because this is the harmony case, cooperators only. And counterintuitively, volumetric productivity is higher in the Hawk-Duff game, the case of coexistence, because this is at higher cell density, because we have to multiply, so to speak, the cell productivity by the cell density. And since this is here higher, coexistence is better. But if you also maximize cell productivity, then this is the optimal point. Okay, there is still some time. Maybe very briefly a third example dealing with pathogenic fungi, namely Candida albicans. And in Jena, there's a lot of research on fungi. Candida albicans is usually a harmless commensal on our skin. However, for example, in immunocompromised patients, it can become aggressive and even life-threatening. For example, here this is picture of candidiasis, which sometimes also occurs after treatment with antibiotics, because it seems that some bacteria suppress the candida, and if the bacteria are killed, then candida takes over. And so, and then if candida enters the body and gets pathogenic, and then macrophages from the human immune system try to attack, or not only try, they attack the candida cells. But this is then really a quite dangerous battle, so to speak, because once a macrophage has ingested candida cells, the game is not yet over, but some of the candida cells then switch from the so-called yeast form to the hyphae form, the multicellular form, and can pierce, also can get out of the macrophage, and finally even can kill the macrophage. But I think there are even chances that the macrophage can win. And and this is an interesting game theoretical problem, because usually several candida cells are ingested, and some of them make this transition to hyphae form and others not. So we called it the piercing strategy and the silencing strategy. And here, so to speak, the macrophage is considered as the environment, and we consider the game between various candida cells. And we again use this concept of playing the field. But now there's interesting mixture, also we, with respect to the population size. Also we consider an infinitely large population of candida cells, but in each macrophage there is a finite population. For example, I think the average number is 7, so each macrophage ingests 7, 5 or 6 candida cells, denoted by lambda, and L is I think it's just the payoff, also the probability of survival. C is the cost for this morphological switch. And then a Poisson distribution occurs, because it could be 1 cell ingested, 2 cells, 3 cells, and so on. And then by some calculation we computed the equilibrium frequency of, these are now the cooperators, in the sense that they attack, also they pierce the macrophage. And the others, so to speak, the factors, because they benefit from the action of the cooperators. Although here, so to speak, the cooperators are in a sense they are the hawks, in the sense that they attack the cell, but they are the doves in the sense that they do the work. This again shows that these terms, hawk, dove, and cooperator, cheater, depends on the perspective. From the human perspective, it's different than from the fungal perspective. And then again, if X asterisk is larger than 1, then all cells fight. In between, it's a hawk-dove game, and if it's lower, then all cells use silencing. And there are also nice correspondence to experimental data, because there are several karyotypes have been observed in these candida cells. Okay. Then some open problems, because here we also for discussing. Namely, an interesting application is sequential cross-feeding, and actually Thomas published several papers about that. For example, I mentioned that some, for example, E. coli cells excrete acetate, and then there can be other cells, actually also other strains of E. coli, who take up the acetate and transform it further to carbon dioxide and water. And this is called sequential cross-feeding, whereas if this gives something back in return, then it's mutual cross-feeding. And importantly, often this intermediate has a negative effect on the first bacterium. It's clear because of the pH, so acetate should not be too large, and therefore it's in a sense also a mutual interaction, because mutualism, because this bacterium also benefits from this one, because this one takes away the acidic acetate. And now this is just the first hypothesis. We have not checked it by experimental data, just an idea that we can consider two different strategies, namely feeding on glucose or feeding on acetate. If both feed on glucose, they have a payoff of let's say 1. But now, if one of them switches to acetate, it depends on the parameters, because usually acetate is a poorer substrate than glucose. But let's assume many cells, and glucose has gone down already, so that it really pays for some of them to switch to acetate. Then let's say they have a payoff of 2, whereas the other one has a payoff of 3. So if one of them switches, it helps the other one more than it helps itself, because the other one then benefits, because then there are not so many cells anymore consuming glucose, so it can take up more glucose. But still there's an incentive for player A to switch to this one, because 2 is larger than 1, although 3 is even larger than 1. And this is the payoff matrix for the leader game. Yeah, in economy there is, for example, a leader and a follower. So if I think this would then be the follower. Yeah, as in sequential cross-feeding, so to speak, the follower. If he switches, then he helps the leader. As the leader benefits even more, but still it pays for the follower to follow the leader, so to speak. And if both use acetate, then it's impossible, because if nobody transforms glucose to acetate, there is no acetate, so they cannot survive. And then we can go even one step further, because we could ask, what is the optimal intermediate? Because it's not necessarily acetate. It could also be pyruvate or lactate, or even earlier in glycolysis, maybe fructose bisphosphate. Okay, the phosphorylized substances probably are difficult to cross the membrane, but let's say there are several possible intermediates. And now the two bacteria have to decide upon the intermediate to be exchanged. And there might be a relation to the ultimatum game we have discussed over coffee. And it's probably a bit far-fetched, but we could maybe think that the first cell is the proposer, so it proposes an intermediate. So it offers acetate and then sees whether the other one accepts the offer and takes the acetate. Because if it does not accept the offer, then the acetate grows up and inhibits this metabolism. So then both of them cannot survive. Yeah? And then the question is, what is the best intermediate to offer? So, and then really now the final slide, because it's more maybe a conceptual problem or conceptual question. What is the lowest level of entities to which game theory can be applied? Because usually it's applied to replicative units like species, populations, cells, genes also, because for example the selfish gene could be considered as a replicative unit and can have different strategies. And then in another paper I found an interesting suggestion, namely to apply it to proteins. For example, in heterodimers, if two proteins bind to each other, there are two strategies, either to be rigid, rigid protein, also mechanically, or a flexible protein. Yeah? And then if both of them are rigid, it's not good because they cannot bind. If one is rigid and the other is flexible, then it's fine because the flexible protein adopts the shape of the rigid one. And this is often observed in these disordered proteins. Also this one, the flexible one is then the disordered one. Whereas if both of them are flexible, then it's not good either. They can bind, but there is no determinate shape, so to speak. Yeah? And then you easily see that it's a hawk-dove game. So the rigid guy is the hawk and the other one the dove. And so conceptually, I think it's applicable, although it's not so clear whether a protein is a replicative unit. Yeah, maybe in relation to the gene encoding it is, but I think this is worth studying further. Okay, yeah, I think this is the conclusion, but I mean this, I have explained all this already. Okay, then thank you very much for the attention. Questions, Thomas?
So, always first to join FBA bashing, but how can one make it better actually? That's a question. What will be next? What's your opinion on that?
Yeah, I mean, one should include kinetic information, because to really describe what's going on, what is the outcome of evolution, even if it's in vitro evolution, kinetics is needed.
They do include kinetics, often for the transport.
Yeah, that's how you get these restrictions, but
But you're right, I mean, if you had kinetics, you wouldn't need an FBA model.
Yeah. I mean, probably a lot can be put into the site constraints. So they usually have, for example, upper limits for some fluxes, for example, the transporter fluxes, and their kinetics can go in, that's true.
Then if you measure kinetics, like you normally do it like outside of the organism, right? So you get whatever rate or whatever you measure, right? And then I don't think that this is really something that's happening in the cell, right? In the cell you have different conditions, and then everything's happening again differently. So,
Yeah,
so
in vivo is often a big difference to in
I
vitro.
don't see so much the point of doing
Yeah.
all this work, taking out system and measuring it in vitro. I mean, it still gives you a good, overview of what's perhaps possible in a way. But this does not give you for sure what's really happening kinetic-wise in the cell.
so how far are we like from maybe metabolomics data to get?
Yeah, as you said already, there are a lot of extensions or the dynamic FBA, and then some including the molecular crowding, so that because the enzymes take some space, and also the should include different costs for enzymes. For example, to make respiratory chain enzymes is much more expensive than to make glycolytic enzymes. And if this is included somehow, then it can be improved. But it takes so much input information that it's not so clear what is then really the helpful output information. What do we then really learn from it if we have to input so much?
Yeah, I think for something like E. coli, it's possible that in 5 or 10 years we'll have alternative sort of full kinetic models for the bacterium, but we'll never be able to do it for the whole of microbial diversity.
Exactly.
Can you explain again? You said that it's not growth rate that is maximized. It's yield. Can you explain why? Why you say that?
Yeah. I don't think I have transparency on that here. Also but I can show something. Also the yield can be written as a ratio of rates. Yeah? For example, let's say ATP production rate, and then usually it's a superposition of let's say stoichiometric coefficients Nij times rates, for example, because ATP can be produced in several reactions. Also, already in glycolysis, it's produced in 2 reactions, and then, of course, in the TCA cycle. So therefore, we must take a superposition. And then for calculating the yield, we have to divide it by the substrate uptake rate and assume for example, there could also be various substrates, for example, glucose and fructose. So in the general case, this is also a superposition. Let's call it, if this is Ni, then it's Nk. So, and what now is usually done in FBA, that they normalize the input rate. So they just say this is equal to 1. This is also written in some papers, sometimes it's tacitly assumed. Per unit input rate, they maximize the growth rate. And then, of course, if this is unity, then this is maximized. And then it looks as if the rate is maximized, because this is a rate, reaction rate. But I think, the crux is really this normalization. Because if we compare respiration and fermentation, then we cannot simply normalize this, because if we fermentation and respiration, if there is now a switch from, actually it depends where the limitation is, and usually, as I said, the rate limitation is in the TCA cycle and in the oxidative phosphorylation. So let's assume we have here a sort of sort of Michaelis-Menten kinetics. So we have here the upper limit, the max. And then respirofermentation actually means that respiration is used up to maximal velocity. However, the limitation here in the glycolysis is much higher. So this Vmax is higher than this one. And therefore now the cell can decide whether it uses in addition fermentation, so that it increases glycolysis, and what cannot be channeled here because there is this limitation goes out here, even without ATP production. Here this is just from pyruvate to ethanol. There's no ATP production, but still it pays to do that because then this can be increased and here ATP can be produced. So here's ATP production and here's ATP production. So, and now in this switch from pure respiration to respirofermentation, the input rate increases, and therefore we cannot normalize this. This is, I think, the thing which is often overlooked in FBA. Yes.